Okay, hello everyone. My name is Ethan, and I'm going to be presenting about on the development of an efficient palladium catalyzed coupling process for Axitinib. So I kind of just wanted to introduce things briefly. So tumors release something known as vascular endothelial growth factor, otherwise known as VEGF. It signals blood vessels to grow around the tumor to provide it nutrients and oxygen. This leads to metastasis and cell proliferation. So this whole process of blood vessels wrapping around tumors is known as angiogenesis. And surprisingly, without angiogenesis, tumors in our body would only grow to the size of the tip of a ballpoint pen. So angiogenesis is implicated in almost all of cancer development. So it's important to find ways to inhibit this from happening if we're to develop effective cancer treatments. So the mode of action, this is kind of just a little bit background on cell biology, but VEGFs bind to the cell walls of the blood vessels. Upon binding, a signaling pathway is initiated. Right here, this red structure, it's called tyrosine kinase, and basically once it dimerizes, which means it forms together, uh, both parts, it releases a signaling pathway. So what Axitinib does, which also the trade name is Enlita, it acts as an antagonist, um, and it inhibits the signaling pathway through basically a growth factor receptor known as vascular endothelial growth factor receptor tyrosine kinase 1 through 3, and that all happens within the cell, so intracellularly. So I wanted to go over the purpose of this experiment, very big picture. So Axitinib, um, the drug has been used for the treatment of renal cell carcinoma, specifically advanced renal cell carcinoma. According to a study that was recently published in Nature, the ORR was 59% with Axitinib in combination with another drug compared to Sunanib, which was only 36%. So ORR is just fancy jargon, but it means objective response rate. And what it means is the proportion of patients in trial whose tumor was either reduced significantly or completely destroyed. So in other words, Axitinib in 59% of the patients with renal cell carcinoma, 59% of them had their tumors either reduced significantly or destroyed. So Axitinib was so successful that in 2019, the Federal Drug and Food Administration, the FDA, approved Axitinib in combination with another drug as first-line treatments for those with advanced renal cell carcinoma. So the purpose of this study, from a process chemistry point of view, was to create an efficient manufacturing process for Axitinib while minimizing the formation of impurities. So the researchers went through a bunch of different schemes in order to officially develop this drug. They landed on scheme 9 as the final scheme. So this is it in all its beauty. But for the next few slides, I'm going to just take you step by step um, through this process. So the same scheme we just saw, but step 1 of the scheme, um, here we see, so the first step was NMP, which is a solvent that you'll see here, was added to a reaction vessel, followed by the product 12, which is 6-iodoindazole. Xanthose, it's a supporting ligand for palladium. It's not always the best to use because it can give rise to a family of impurities, but it was successful in this case. And then dipalladium was added with sodium bicarbonate. So sodium bicarbonate here is a milder base. It was used instead of cesium hydroxide, which was used in a previous scheme, and it allowed the Magida coupling reaction, which is these two compounds coming together to form 13. So it allowed that to occur at much lower temperatures. So um, The reaction was next purged with dry nitrogen. The mixture was held at 25 degrees Celsius and stirred for 30 minutes. So a separate solution of compound 10, which is the thiol, was added over 1 hour. But it's important to note to combat inhibition of the palladium catalyst, which occurred frequently, 10 needed to be added slowly to the reaction mixture. It was also important to not keep 10 alone with the palladium catalyst, or it would react and inhibit it. So this batch was heated to 50 degrees Celsius and held for 2 hours. When less than 5% of compound 12 was remaining, step 1 was considered complete, and the mixture was cooled down to 25 degrees Celsius. So I think it's important to take one step back and ask, why do we use dry nitrogen? Why was the reaction vessel purged with dry nitrogen? And the reason is product 10, which is here, can combine with itself, which is known as dimerization, to form product 15, which is called a mercaptodimer. And this happens if oxygen is not excluded from the reaction. And then once product 15 is formed, it can react with palladium via oxidative addition to create product 16. And that process is problematic because it inhibits the palladium catalyst, which is needed further down in the reaction scheme. So moving along, this is step 2 of scheme 9. So a solution of iodine, I2, and NMP was added while maintaining 25 degrees Celsius. Then a solution of potassium hydroxide in 40 liters of water was added over 30 minutes while maintaining 25 degrees Celsius. So low temperatures was really important so as not to develop a sulfoxide impurity. So the impurity that they identified, they labeled it as number 18, and a sulfoxide impurity meaning S bonded, double bonded to O up top over here. So this mixture was allowed to sit for 5 hours. Once no more than 7% of 13, compound 13 was remaining, step 2 was assumed to be complete. So the reaction needed to be stopped, so it was quenched or stopped by pouring it into a solution of ascorbic acid in 120 liters of water over a period of 1 hour at 45 degrees Celsius. So 120 liters of methanol was added to the slurry as well. It further helped stop the quenching, or it further helped quench the reaction to stop excess iodine from reacting. Um, so the suspension or the mixture was stirred for 3 hours at 45 degrees Celsius. Then that slurry was filtered while kept warm. So the filter cake, which is the solid in the funnel, it was washed with 120 liters of water and then 120 liters of methanol. So once dried, a brown crystalline solid was produced, and it weighed in at 62 kilograms, and it had a 92% overall yield. So step 2R was the recrystallization process of product 14. So recrystallization is a purification technique for solid compounds. Here it was used to remove the xanthose related impurities from the previous schemes, as well as other impurities that came up. So the reaction vessel was first purged with inert gas, which is that nitrogen we talked about. To it was added 62 kilograms of the crude and impure 14, then NMP, and then 1,2-diaminopropane. So 1,2-DAP, which is 1,2-diaminopropane, was added to remove color and help solubilize palladium to isolate it later, because it later will need to be removed. So the solution was heated to 60 degrees Celsius and stirred to dissolve the solid. Then 338 liters of methanol was added while maintaining 55 degrees. 335 liters of water was added next while maintaining 55 degrees Celsius. So water and methanol were added as what is called anti-solvents. So something the compound is less soluble in, so we're able to isolate it later. So a slurry resulted. That slurry was allowed to cool to 20 degrees Celsius, and then it began to crystallize or granulate for 3 hours. So the resulting slurry was filtered with methanol, water, and it was blown dry with more nitrogen. So this resulted in a pale yellow crystalline solid weighing 45.3 kilograms with 73% yield and 99.5% purity. It also had a melting point between 219 and 221 degrees Celsius. And to confirm that 14 was indeed what was synthesized, an HNMR was taken, um, just to confirm that 14 was indeed present. So now I'm gonna go through steps 3 and through 5, but just on this slide, I'm gonna talk about steps 3 and 4, and we're gonna save step 5 for the next slide. So the purpose of steps 3 through 5 was to produce 1C, which you'll see right here, which is a crude form of Axitinib. So it's a further synthesized form of Axitinib. So steps 3 and 4. Okay. So the researchers, it was a little strange because in the literature paper, how they performed this reaction didn't necessarily line up with what's written in this reaction scheme. But I'm just gonna go through what the researchers did. So first, palladium with acetyloxy groups attached. So 2 acetyloxy groups were added, um, as well as xanthose and NMP to a nitrogen purge reaction vessel. So the palladium officially couples 6, which we talked about earlier, um, without generating this specific impurity. So an impurity that cropped up was impurity 26. And if you'll look here, the goal was to create a trans alkene, but the impurity that came up without the use of xanthose was just an alkane. So it removed the double bond. So it also proved important to balance xanthose and palladium catalyst, specifically in a 1-to-1 stoichiometric ratio. So what they found was that having too much xanthose led to the inhibition of the palladium catalyst, and then too much palladium catalyst, on the other hand, increased levels of impurity 26. So next, a base known as Hunning's base, which is an amine, was added, as well as 14, which you'll see here. The reaction was heated to 50 degrees Celsius, and then acetic anhydride was added to cause acylation. So you'll see that here. The acyl group was added. So the acyl group was important. So it's known as a protecting group. So otherwise, 6, which is pretty much this compound here, which is attached via the Heck reaction. So without the protecting group, 6 would just, instead of forming down below like it's supposed to, as you see here, it would form up top, which is a problem. So also the inclusion of the Hunning's base allowed the Heck reaction to be at 90 degrees Celsius rather than 110 degrees Celsius, which is important because it just makes the process chemistry a lot easier, a lot more efficient. So Hunning's base was also proved really critical because other bases had much lower conversion rates. So other bases didn't convert to the product that we needed, or it took much longer for that conversion to take place. So finally, once no more than 2% of 14 was remaining, it was just assumed that the acylation was complete. And once complete, product 6, which is 2-vinyl pyridine, was added. So they also found that there needed to be like an exact equivalency. So too little of the vinyl pyridine was a problem. Um, so adding 4 to 8 equivalents actually lowered the number of impurities produced. So after the vinyl pyridine was added, the heating was increased up to 90 degrees Celsius for the Heck reaction to take place. And the Heck reaction is just what allowed the vinyl pyridine here to be added. So once no more than 2% of 29 was remaining, 29 right here, it was assumed that the reaction was complete. So next, the reaction mixture was cooled to 50 degrees Celsius. So now I'm going to go through step 5 on the next slide. So here's step 5. And we see product compound 30 here. So the mixture was diluted with THF, which is tetrahydrofuran. So it was passed through a polishing filter. So THF is a good solvent that can dissolve lots of different products, even at room temperature. But importantly, it was critical for this reaction because it enabled deprotection of the acyl group. So it allowed the acyl group here for us to get rid of it. It also helped to solvate any palladium that might have been remaining in the reaction vessel. So further dilution took place with more THF and 1,2-diamino propane, which is the 1,2-DAP we see here at 50 degrees Celsius, and it was stirred for 30 minutes at 50 degrees Celsius. So after the stirring, 496 liters of water was slowly added while the temperature was maintained at that same temperature of 50 degrees Celsius. The mixture was stirred for 12 hours, and then finally, after the 12 hours, it was cooled down to 15 degrees Celsius. That slurry, and since it was cooled down, the filter cake, which is the solid, was washed with water. The crude material was again made into a slurry by adding THF with 1,2-DAP for 4 hours, so that helped dissolve it. Then it was cooled once again down to 15 degrees Celsius. Then it's filtered. Then it was filtered one more time, washed with THF, and dried. So the final product that resulted was what we see here, 34 kilograms of it, and 80% yield with 99.3% purity. And it had a melting point of 216 to 127 degrees Celsius. So to confirm no residual palladium from step 4 was hanging around in the reaction mixture, or in the solid rather, an atomic absorption spectroscopy was performed, and that was able to successfully determine that no more than 20 ppm, parts per million, of palladium was present as an impurity. So scheme 9, step 6, so this is the final step. So it was the final recrystallization process and polymorph conversion. So polymorph conversion is a fancy name. It's also known as a polymorphic transition. But basically, it's a transition from a solid crystalline phase to another phase with the same chemical composition, just with a different crystalline structure, crystal structure. And the goal was to form compound 41. So executing proper polymorph transitions is important, especially in the pharmaceutical industry, because some drugs exist in different crystalline forms, which differ in their physical properties. So first, a reaction vessel was purged. To it, 1C was added here, then NMP and THF as the solvents. The reaction was purged again with dry nitrogen, and the mixture was heated to 68 degrees Celsius and stirred to allow dissolution. In a separate vessel, ethanol was added, and the reactor was heated to 73 degrees Celsius and stirred. The NMP, THF were essential because they completely dissolved Axitinib. Then it was transferred to a separate vessel with the ethanol. So So to this hot solution, seed crystals of 1E were added. Seed crystals are used as a base to grow more crystals in much the same way seeds help grow plants. So the heating was increased so a reflux could occur, and a reflux involves heating a reaction to boiling temperature and allowing the solvent to recondense into the reaction flask. It's a simple way to maintain a constant reaction temperature. So then it was held for reflux for 7 hours. After the 7 hours elapsed, the mixture was allowed to slowly cool down to 20 degrees Celsius. The slurry was then filtered and washed again with ethanol to get rid of any mother liquor or any NMP. So then 1E was allowed to dry. It produced a white crystalline solid weighing 28 kilograms with 82% yield and 99.9% purity. So at this time, the palladium level was once again analyzed using atomic absorption spectroscopy, and it was determined to be no more than 6 parts per million. And an HNMR was also determined to confirm this product. So I just want to conclude very briefly here. So this experiment was important to develop an optimized manufacturing process for Axitinib, which is an angiogenesis inhibitor used in cancer treatment, specifically renal cell carcinoma treatment, specifically advanced renal cell carcinoma. So the researchers developed this method, ultimately landing on scheme 9, which involved 2 palladium-catalyzed coupling reactions, a Heck reaction, and a Nagita coupling. In the process, a few hurdles were encountered, such as the development of 5 impurities resulting from the Heck reaction, 6 impurities from steps 1 through 2, as well as several low-yielding steps. So those needed to be overcome. But once scheme 9 was settled on, it was optimized in only 6 steps with an overall 50% yield. So that's all I have. Thank you very much for listening. I really appreciate it.